Okay, this is chapter 17 from the book, The Medical Reformation. This chapter has a lot of stuff. We're going to break it into several different lectures, at least 3 different lectures. We're going to start out by talking about how a neuron plasma cell membrane works and the concept of excitotoxicity. We'll do the separate parts later, in parts A, B, and C. This is really useful stuff because this explains how to understand hypertension, how to understand dementia, and just basic cell physiology. So these concepts, it'll be real clear. You can hit the print screen button if you want to save these drawings as a slide, but you really want to understand this. This is fundamental to understanding dementia, to understanding hypertension. Okay, so every cell uses these plasma membrane pumps. K is for potassium, like kalium, the Latin word for potassium. N is for sodium, for natrium, the Latin word for sodium. So this is a neuron, brain cell, and it has a negative 65 millivolt plasma membrane gradient. And what that relates to is the electrical charge. It's more negative inside the cell than just outside the cell. It also has what is called a chemical gradient, meaning that sodium is more concentrated outside the cell than inside the cell. And because of that, sodium wants to come into the cell. It wants to come into the cell because its positive charge is attracted to the negative charge inside the cell. It wants to come into the cell just because ions naturally want to equalize their gradients, meaning be the same concentration inside and outside of the cell. And that desire of sodium to come into the cell is then coupled to doing other types of work. It'll be coupled to pumping calcium out of the cell. So this pump right here is called the NACA exchanger. Na for sodium, Ka for calcium. It's a lot of times abbreviated NCX. This pump right here is actually the most important one. This is the potassium sodium ATPase pump. And it uses ATP, which is the energy currency of the cell. This is what's made in the mitochondria. ATP means adenosine triphosphate. It requires energy to pump the sodium out of the cell because you're pumping against the gradient. Sodium is more concentrated outside the cell than inside the cell. When you pump against a gradient, you have to use energy in order to be able to do that. And about 50% to 2/3. You can think of it as being 65%, just like the membrane potential, negative 65%. Think of that as being about 2/3 of the energy of the cell. About 2/3 of a neuron's energy will go to running this pump. So anything that would take so much energy for the cell must be super important, and it is. And so the reason is neurons have to do a lot of work of pumping things in and out of the cell. So in order to generate a gradient, the neuron invests tons of energy into this because this is like electrical power inside a cell. It's like a battery in a sense that you store charge on 2 sides of a charge barrier, and then you can have current flowing, and the positive ions can flow in and out of the cell. Okay, so what's going to happen is by constantly pumping more sodium out of the cell, it pumps 3 sodiums out while pumping 2 potassiums in, you're building up more positive charge outside the cell than inside the cell, so you have a negative resting membrane potential. You're also building up, like I said, a charge gradient with higher concentrations of sodium outside the cell. And that's going to be real important because you need these gradients in order to be able to handle calcium. Sodium and potassium are like minimum wage workers. They work their butt off all day long, okay? But really the aristocrat of ions is calcium. Because when calcium gets increased in the cytoplasm, that makes something happen. It's the boss. Comes in and says, do this, do this. Okay, so calcium can be thought of as like a light switch to turn the cell on and off. When calcium concentration raises in the cell cytoplasm, something happens. Whatever that cell most importantly does, that's the thing that'll happen when calcium comes in. So that's going to be key to understand. So I don't know if you can see this light, like a light switch, on and off. That's what the effect of calcium is in a cell. Okay, by the way, in order to get these pumps to run normally, a person needs to eat a lot more potassium than sodium. That's what we are designed to do. We're designed to eat probably at least about, some people say you can get by with as low as 5 to 10 times as much potassium as sodium, but our ancestors probably ate more in the likes of about 20 to 25 times more potassium than sodium. Okay, whereas modern Americans or Westerners have reversed that. They're tending to eat about, 10 or 15 times more sodium than potassium. They're doing the opposite of what they're supposed to. And that will cause problems with their plasma membrane ion gradients and will cause a decrease in the function of their cells. Okay, plants basically have all the good stuff. So like I said, I don't have any other, anything else about this vegan diet except that it works. It's what humans are designed for. And basically, the more you study it, the more obvious it becomes. Plant foods basically have all the good stuff. They got more fiber, which maintains your gut barrier so you don't get leaky gut, and maintains your blood-brain barrier to protect your brain from toxic chemicals that are in the blood transiently after eating meals, for example. More potassium, which are vasodilators. More magnesium, which are vasodilators. More nitrates, which are the precursors to make nitric oxide, vasodilators. Okay? More antioxidants to prevent problems like oxidative stress that'll damage cells. More alkaline, so it protects the kidneys, protects the bones. When a person eats excessive amounts of sodium, as most Americans do, the kidney will automatically start, urinating out potassium. And this can lead to diminished levels of potassium over time. The body has to maintain, balanced amounts of cations. Cations are positive ions like sodium and potassium. So if you're taking in lots more sodium, you're going to excrete more of your potassium. And when that's overdone, it can start affecting plasma membrane gradients. Okay, you can basically, cause a problem for your cell battery, your cell's electrical system. Okay. Basically, like a skeletal muscle cell, a vascular smooth muscle cell, when calcium goes up in the cytoplasm, it will contract. A neuron, the main thing it does is release a neurotransmitter, so it will release its neurotransmitter. Okay. I'll also explain, let's see here, let's get to a couple of these things. All right, now I'm going to show how a neuron works in more detail. So here's what we just talked about, the potassium sodium ATPase pump, taking in 2 positive charges, 2 potassium, and pumping out 3 sodiums. So building up a negative charge gradient, also building up a concentration gradient. Sodium is about 10 times higher outside the cell, 140, compared to inside the cell, about 14. And then when you want to pump calcium out of the cell, again, you have to pump against a very high gradient because calcium concentration outside the cell is about 15,000 times what it is in the cytoplasm. So that's an incredibly high gradient. So that's why you definitely need a lot of energy. By the way, the potassium sodium ATP pump is called primary active transport using ATP. This knockout exchanger is called secondary active transport because even though it doesn't use an ATP as part of its direct pump action, it's based on the gradient produced by ATP used over here at the potassium sodium pump. Okay, so this is just showing excess calcium in the cytoplasm, the neuron will release its neurotransmitter. Okay, if you have excess calcium inside the smooth muscle, it'll contract. Okay, and then this gradient of sodium, electrochemical gradient, is also used for doing other work, like for pumping in amino acids, for pumping out acid, pumping out protons. Okay, it's the electrical power system of the cell. One of the things when you study cell physiology, you start to realize is the cells are like chemical electrical geniuses. They know how to do all kinds of stuff that humans are just beginning to understand. Okay, we talked about the gradients. The NCX, knock exchange, is a really fast pump. So most of the energy for the work done in a cell comes from the mitochondrial production of ATP and then from secondary utilization of these plasma membrane pumps. Okay, here just shows a little bit about ion concentrations. Again, sodium's about 10 times higher in the ECM, extracellular matrix, compared to the intracellular part of the cell. With potassium, it's about the opposite. It's about 10 times higher inside the cell than outside the cell. And calcium's, 10,000 to 15,000 times higher in concentration outside the cell. Okay, that's why we always got to pump against the gradient. Okay. Okay, all right, now we're going to look at, a neuron conducting an action potential. First of all, just the anatomy of a neuron. Here's the cell body, and that's where the nucleus is with the DNA. The nucleus has what are called dendrites. Dendrites, like the Latin word for tree, so they look like little trees, and that's where they receive signals. And when enough signals come in to activate the neuron, make it fire an action potential, the action potential starts about right at the end of the cell body. That's called the axon hillock. This elongated tubular structure right here is called the axon, and that is for sending the action potentials, the nerve impulses. The neurons are typically coated in a type of fat that's called myelin. And what it does is it speeds up conduction because the conducting neurons can jump between these blobs of fat, myelin coating segments, and the uncoated segments are called nodes of Ranvier. And so with the sodium action potential, the conduction is going to jump. It's called saltatory conduction, to jump through these nodes of Ranvier until it gets to the synaptic terminal, sometimes called the axon terminal. And I'll show you in the next picture what happens. But these are neurotransmitter vesicles. When activated, they will travel to the plasma membrane and they will, of the presynaptic neuron. They will then be merged with the membrane. The neurotransmitter is released, diffuses across the synaptic cleft, and then it binds to the postsynaptic neuron's receptor, and it will exert an effect on the postsynaptic neuron. Okay, here's the same drawing in a little more detail showing you what's happened with an action potential. Sodium channels will open up. Sodium comes in, and that's the main ion of importance for the action potential. Once it gets down here to the axon terminal, it will have depolarized the cell. This will open up the calcium channels. Calcium will then flow into the synaptic terminal, and the increase in calcium within the cytoplasm of the axon terminal will cause these neurotransmitter vesicles to travel to the plasma membrane of the presynaptic neuron and then to release their neurotransmitter. As they merge, they'll just merge and their neurotransmitter will be pushed out in the synaptic cleft. Again, it binds with the postsynaptic neuron, exerts an effect. Okay, and so that's, remember too, it's the voltage that opens up those calcium channels, because there's going to be other things that open up those calcium channels. Here, I'll just look at it again. So I could have put more detail on there, called these VGCC, voltage gated calcium channels. So when the voltage comes up, because of all the sodium depolarizing the cell, changing the charge in there, that will open up the voltage gated calcium channels, and they will then exert their effect. So we're going to leave it at that for now. When we come back so this will be the end of chapter 17a. When we come back to 17b, we're going to go how this relates to psychiatric medications, and then we're going to go into go into how this relates to causation of dementia, and we're going to go through the neurovascular uncoupling theory. That's my theory of dementia. So we're going to get into some really interesting neurophysiology. And by the way, this is more neurophysiology than you'll find in any book. I've gone through all these books about Alzheimer's, and they don't explain this. And this is really basic stuff. And you have to understand this in order to understand the excitotoxin theories. You have to understand this to understand EMF. You have to understand this to understand neurovascular uncoupling. So that's the key point. Sodium channel initially conducts the action potential, gets to the axon terminal, opens up voltage-gated calcium channel. That activates the neurotransmitter vesicles to travel to the plasma membrane, merge with the plasma membrane, release their neurotransmitter into the synaptic cleft. Neurotransmitter diffuses across the synaptic cleft, binds to a receptor on the postsynaptic neuron, exerts an effect, either excitatory like with glutamate or inhibitory like with GABA. Okay. So anyways, we will go through the rest of this in the next lecture.